It was almost exactly 2 years ago. I got a call from Charlie Izzard, who's on the board of Tract. John, would you be willing to read a few scientific papers for me? The next day I called him back and said, send me more. This is fantastic science. It's some of the most exciting science I've seen. Charlie, it's almost too good to be true. I think this will change the course of cancer research in the country.
My name is Frank McKeon. I'm a co-founder of Tract Bio.
My name is Hua Xian. I am the co-founder of Tract.
An increasingly broad range of chronic inflammatory diseases, a broad range of cancers. They're widespread. By the time someone is 60, there's a good chance you have one of them, and we don't have good therapeutics for these diseases. So our goal is to sort of crack that code.
As I got to know Frank and Hua, they had found something that no one else had found, and that is that certain inflammatory diseases that are serious and common, and certain diseases that can turn into cancer, all shared a common element, which appears to be a stem cell, that is to say, an immortal cell that actually can cause those diseases.
The stem cells are the seeds of the disease.
The ability to grow these stem cells up so that you have literally hundreds of millions of these cells in a pure culture, we call that cloning. And no one has found a way to clone epithelial stem cells except this pair.
We developed this technology platform, and we can use it to clone stem cells from nearly all types of tissues in human body. This stemECHO technology can allow me to clone these stem cells and expand them from one single cell to 1 billion cells in about 6 weeks.
Once you do cloning, you realize, well, actually half of those cells are good, and the other half are bad. If we killed the bad ones, you would have the good ones. Now you have a clone cell that you can screen against 10,000 or 100,000 different drugs in parallel with the normal clone stem cell, and it becomes very easy to do drug development.
This is our new approach of drug screening, and by using this approach, we identified a drug combination that can selectively kill pathogenic stem cells and cancer stem cells without hurting the normal stem cells in our body. We believe if we eradicate cancer stem cells, we can cure cancer.
The dominant technology in the field is called organoids. We started to think about it when we got samples from Crohn's disease. If you used organoids, you would come to the conclusion that the entire gastrointestinal tract was defective. When you use stemECHO, you generate a series of clones, and you realize some of those clones are completely normal, and some of those clones are disease-causing. And that's the fundamental concept.
Hundreds of millions of identical cells that are immortalized. They will live forever in the lab, and you can study the science, find treatments, find diagnostic markers, and so on.
There's a common theme for chronic inflammatory diseases such that we could go after, and we are going after, a number of others, Alzheimer's, and we fully expect them to be operating in the same way.
We have opportunities in diagnostics as well. For instance, we've already shown that we can take cells from biopsies and we can actually grow them up within 10 days and test them against drugs that are currently available to understand which drugs to specifically treat that patient tumor with. Another big opportunity for us is in regenerative medicine. One of the things that we could potentially do with the stemECHO platform is isolate individual stem cells from organs, diseased organs, isolate out the healthy ones, grow up healthy organ tissue, and replant it back into patients.
What Tract will do is resolve the basis for these widespread chronic inflammatory diseases that we don't have treatments for.
Our immediate priority is to focus on research and development. And we're really hyper-focused on discovering new drugs that selectively kill these stem cells. And I firmly believe that what we're doing here at Tract is going to fundamentally change the treatment landscape in cancer, inflammation, regenerative medicine, and diagnostics.
Some patients, they do reach out to me and Frank, and we kind of feel hopeless because we tell them we are just researchers in the lab. But Frank always said, my research could help more people than MD, because if I have drugs and I can push to clinical trials, and there will be millions patients could benefit from it.
The more scientists look at this across other diseases like inflammatory bowel disease, ulcerative colitis, Crohn's disease, possibly Alzheimer's disease, possibly brain tumor, there's an awful lot of applications that will come from this work. It's also an opportunity for people who want to have impact and want to support science and help it move more quickly. The impact that this work could have is immeasurable, and I think it would be very fulfilling to be part of accelerating this kind of work.